Okay, yep, I think we're good. Right. Well, welcome to the final session. I'm not Julian. My name's Richard Ivanovic. I'm also from Liverpool. And it gives me great pleasure to introduce Stella Mazeri, who will hopefully appear on screen. He looks across, hopefully, at the AV. Excellent. Okay. And I can confirm. Yep, you are showing your slides, Stella, so if you wish to take over.
Hi there, I hope you can hear me as well.
Yep.
Brilliant. So thank you very much for inviting me to talk today, and sorry that I had to do this online. Thanks for the great talks we've had already, and I know this is the end, so I'll be brief, and the presentation will be quite simple and straightforward. Right, so I'll be basically just taking you through what we know so far on epidemiology of the female coronavirus outbreak in Cyprus and our future plans. So in this talk, I will describe the main data sources that we have available to describe the outbreak, describe the evolution of the outbreak in space and time, tell you a bit about the characteristics of the outbreak in terms of the different forms of FIP and characteristics, and I'll finish by mentioning the future work we hope to carry out in Cyprus, Greece, and beyond. So we have two main data sources that describe this outbreak. The first one is being collected by Vet Diagnosis, veterinary pathology lab, who is the main provider of FIP diagnostics in Cyprus, and who were the first to document the massive increase in cases. This dataset provides data on individual cats who are confirmed to be FCov positive, either through PCR and/or immunohistochemistry, and the data is regularly updated as samples come in and get tested. The second source of data comes from an online questionnaire administered by the Pan-Cyprian Veterinary Association, and this was administered in December 2023. And the main aim of the questionnaire was to capture crude figures of cases per trimester in 2022 and compare that with 2023 and give us an estimate of the overall burden. So both data sources have strengths and weaknesses, and I'll just briefly discuss those now. So for the laboratory data, we have a total of 209 samples, so obviously it's not a very large data set, but these are collected in real time and is the only source of confirmed cases with individual cat data that we have. Nevertheless, based on the questionnaire data, only 1/3 of the vets reported that they might use PCR to confirm diagnosis. And we estimated that the total number of cases during the 2022 and 2023 was around 8,000, with the majority of those, of course, being during 2023. On the other hand, the online questionnaire was completed by 40 vet clinics, which is around 1/4 of the clinics in Cyprus, and they report around 2,000 cases during this time. So while this questionnaire may provide a more complete picture of the outbreak, it suffers from granularity and potential recall bias. Due to the nature and timing of this questionnaire, data was collected as crude numbers of cases per trimester without confirmation, and no individual cat information was available. So today I will mainly focus on the results of the laboratory data, since we have individual cat data are collected in near real time and represent confirmed cases. So you probably have seen this version of this plot already, but just a quick recap. In the early days of 2023, we noticed a really high increase in FIP cases, which was more than 20 times of what we had seen in the whole of 2022. We think it started in Nicosia, shortly followed by Limassol, and by March it was actually all over the districts. So according to the data, the outbreak peaked in spring 2023, and even though we saw a decrease in cases after that, cases remained high during the summer. Cases appear to be reduced during the winter 2023-24, but importantly, there are still confirmed cases in every district, and we have recent reports of vets describing a new increase in cases during this month. This just reminds us that it's very important to continue monitoring the number of cases and the spread and be able to anticipate treatment needs and understand the risk of spread within Cyprus, but also beyond Cyprus. So in order to check how the two datasets compared, we aggregated the laboratory data by quarter to be able to basically compare them together. So if you see these two plots, the top being the questionnaire, the bottom being the laboratory data, we see that they both describe a large increase in cases at the beginning of the 2023 compared to the previous year, and the cases remain high for the duration of 2023, even though there is a bit of a decrease, especially in the laboratory data set. They're still much higher than what we've seen before. There's still sort of reasons to worry. This sort of agreement between the two data sets reassures us that the outbreak described is likely to be real and that there's value in both data sources. So in terms of clinical forms, FIP forms, the most common one was the effusive or the wet form, which was 65%, followed by the neurological form with 28%. And then the dry form with 7%. And while in the wet form is the most common form with classical FIP as well, we normally see much fewer neurological cases. So this could be either due to increased awareness of the presentation, because there was a campaign to increase awareness of FIP in general in Cyprus during this outbreak, or it could be an inherent to FCov23 neurotropism. So this is something to look into. Then in terms of the cats characteristics, the median age of the cats who developed FIP was 3 years of age, with the average being around 5 years old, and ages ranged from 1 to 19. So technically cats of any age can develop FIP, but it's actually much more common in cats under the age of 2, especially cats under the age of 1. But here we see older cats. We see cats between 2 and 8 years old to be affected, which is quite unlikely and uncommon in the classical form of FIP. Similar to the classical FIP, though, male cats were overrepresented in our dataset. In terms of housing status, cases were more common in free-roaming cats or cats that lived in cat shelters, which is to be expected, followed by cats were allowed to be outdoors. But interestingly, we had around 5% of cats who developed FIP were indoor cats. Cats that were not allowed to go outside, which raises question about how the virus is transmitted. So the recent FIP outbreak in Cyprus was unprecedented, spreading very fast through the island, infecting even indoor-only cats, cats of all ages, and causing increased proportion of neurological FIP. The great work by colleagues at the University of Edinburgh has demonstrated that the majority of cases are likely to be caused by the new FCov23 strain, which is likely the reason for the different nature of the outbreak we have seen, but there's a lot we don't know about it. So far, the funding to study this has been limited, and I think my colleagues have reiterated this enough. So a lot of this work was actually voluntary and happened in our free time, etc. But thankfully, this outbreak was also the reason for the development of the FCov23 Center of Excellence, through which we aim to run a number of complementary research projects to try and gain a better understanding of the different aspects of FCov23. So today, I'm just going to briefly introduce the epidemiology projects, but of course, there's a lot more in the pipeline, some of which my colleagues have already discussed. So the first piece of work will focus on the epidemiology of FCov23 in shelters in Cyprus. We aim to recruit a representative number of shelters and monitor for any new cases with the aim to understand the risk within and beyond Cyprus. Additionally, we will try to better understand how FCov23 is transmitted through different sampling types, for example, eyes, nose, saliva, and feces, as well as environmental sampling, such as cages, litter trays, feeding bowls, water bowls, but also clothes and shoes of cat carriers and cat transport vehicles, to understand the potential of mechanical transmission as well. We will use laboratory methods developed by colleagues at Edinburgh, including multiplex PCR, which will enable us both to detect infection, but also to identify the strain. And we're hoping to also use virus isolation to check whether the virus survived in the different sample types. And this work will hopefully identify potential control measures at the local and international level. But importantly, we're going to be in close communication with the shelters where a lot of it will happen. We will also use samples submitted for PCR via the vet clinics to continue monitoring the clinical cases. the clinical manifestation, and the virus strain. And for both of these studies, we will sequence a subset of cases to continue monitoring the evolution of the virus in Cyprus. Lastly, we will also carry out research in Greece due to the strong links between the two countries and the strong similarities, as well as the large stray population in Greece. We will collaborate with first opinion and referral companion animal practices in different locations in Greece to test FIP clinically suspected cases. This will help us detect infection and identify FCov strain to estimate the burden in Greece and understand whether the recent increases, there's been anecdotal increases in Greece of FIP are due to the same strain or another strain. We will also try to identify potential risk factors using sample metadata from accompanying questionnaires from positive and negative cuts from the clinics. And again, we will sequence a subset of samples to understand what's really going on and try to link the sequences from the different places. Lastly, we will use the data from all these studies to carry out quantitative risk assessments for the introduction of FCov23 to other countries from both Greece, if we find it's there, and Cyprus. So of course, there's a large team behind this project, and a lot of this work is yet to be done. You've met a lot of my colleagues already, but just to make sure, I would like to thank our team in Cyprus, Greece, Edinburgh, and the FCov23 Center of Excellence, as well as everyone who has supported this work or will support this work in the future. Thank you again for inviting me to give this talk, and everyone for your attention. Thanks very much.
Okay, thank you very much, Stella. If I can open the floor to questions. Just whilst the microphone is moving round, if I can have a cheeky one to start with. The samples that you're collecting, are they going to be blood samples, fecal samples?
So we will collect, so for example, the owned animals at the clinics will be just your typical FIP samples, but we're additionally collecting fecal samples, saliva samples, samples from the eyes and nose, just to check whether we can find the virus there as well as the environmental sampling. Cool, okay.
Will those samples be kept, or are they just going to be used and then disposed of? As in, are you setting up like a biobank of these?
Yeah. So as part of the consortium, there's going to be a biobank prepared as well. Yeah.
Cool.
Thank you.
So I know I could ask this question to you as well in person, but I was wondering, with the recent uptick, does the kitten season start a bit earlier than April in Cyprus, or when are most of the kittens, the first kind of seasonal breeding, born in Cyprus?
I might get Haralambos to answer that, or Dimitris is online.
Yeah, I will. I'm online.
Yes, Dimitris.
You hear me?
Yeah.
Yeah, thank you, Christy. Question. Yeah, the season has already started. We see the mating season is starting in January in Cyprus. So we probably have the first kittens in March. So yeah, it's already started.
By the time we start collecting data, they'll already be infected.
Yes,
but
yeah.
Hopefully we'll be collecting data for 2 years as well, so we should catch another season as well.
Excellent. Okay. Yep. There's one more at the back.
Hi. Thank you for the great work. I'm a first-line vet. So I cannot comprehend how there was this explosion of the virus in all of the island. Okay, do we have any idea how it did spread?
We think that it's potential. People are potentially involved, not by getting infected, but potentially carrying the virus on their clothes and shoes, which is why we're testing it. It's a bit strange that we have indoor-only cats affected, for example. So there are a lot of unknowns in this virus. We can only make hypotheses, but hopefully during this work we'll find out more. But yeah, you're right. It spread quite quickly throughout the island.
And another question: are is it in your plans to put a more like specific map of the cluster cases that we see, like more pinpoint, like on a GIS system, and see what is happening, depending on the population density?
So we have information about the location of the approximate location of cases in the smaller data set. So we might, if we get ethical approvals to show that, we might. It won't be exact cases, but yeah, definitely it will give us a bit more granularity on what happened. But bear in mind that there's cats everywhere in Cyprus, and this is going to be a small subset of what really happened.
Very nice presentation. Thanks a lot. I was wondering, in your studies, do you also include serology? So the question is, so will there be population immunity at some point, and would that help?
As far as I know, with FCov, the immunity doesn't last long, so it might give you a drop, but it won't. As far as I understand, cats can be reinfected quite soon after. Don't know if anyone in the audience knows more. So I think, and no, we're not planning to do serology, at least in the shelters. We're trying to do non-invasive sampling, but we could do in the animals.
We're definitely going to do serology on the cats that are recruited via the laboratory path, because we have a leftover serum from these cats. We haven't yet tested them, but it's something we plan to do. And we also have archived samples from 4 years ago, before the outbreak. We're also going to test which feline coronavirus were circulating in the island prior to the outbreak, at least from serological perspective. So it's definitely something we plan to do.
And I think it would be interesting to look at serum from not just from the FIP animals,
absolutely,
but in general.
Yes. So we have a control
even during the last year.
What I'm going to say is for information, really, with regards to potential transmission of the virus between aggregations where the cats are outside in the community. There are volunteers who go from station to feed the cats, and they clean their eating utensils, they put out water bottles, they put them in cages, and they take them to vets to be neutered. So these people can act as potential transmitters of the virus from station to station. So this is what's happening, not just in Famagusta where I live, but in Nicosia I see them going round with bags of dry food to feed the cats.
Yeah, absolutely. There's a lot of cat lovers in Cyprus, which sustains the population, and they might be involved in transmission.
Okay, I think we've exhausted the questions. So if I can say thanks again to Stella for joining us online. That was a fantastic talk.
Thanks very much.